Today, I'm going to teach you about MicrobeTrace. For those who may not be familiar with MicrobeTrace, it is a data visualization tool that allows you to display both genomic and epidemiological data simultaneously for outbreak investigations. So before we dive in, I'd like to review the learning objectives for today's lesson so you know what to expect and what you should be able to take away by the end of this presentation. After participating in the session, you should be able to describe what MicrobeTrace is, its role in public health, data visualization, explain the key functions and capabilities of MicrobeTrace, define transmission networks and their relevance to outbreak investigations, and lastly, identify the types of data and information required to use MicrobeTrace effectively. So now let me give you a bit more detail about what MicrobeTrace is. It is a powerful secure data visualization tool that can be used to map transmission networks. However, transmission networks are not the only things MicrobeTrace can visualize. It can also display histograms, SNP heat matrices, nucleotide sequence alignments, phylogenetic trees, and maps showing geographic locations of the uploaded cases. Because MicrobeTrace supports these different visualizations, it's able to handle a variety of data inputs. Also, MicrobeTrace is pathogen agnostic, meaning it can be used for any pathogen capable of causing an outbreak. While it was originally developed for HIV surveillance, it has been since used across a wide range of diseases. For example, I use it frequently when collaborating with the healthcare-associated infections team for outbreak investigations regarding Carbapenem-resistant organisms. The best part is that MicrobeTrace runs entirely within your web browser and does not transmit any information over the web, which is critical for maintaining data security and HIPAA compliance. Transmission networks provide fine-grained insight into transmission patterns by visualizing connections among individuals within a group. These networks can be epidemiological or based on genetic distance. MicrobeTrace allows both network types to be visualized simultaneously, enabling integrated interpretation of genomic and epidemiological data. So, why do we map transmission networks? First, they help us identify where transmission is happening, allowing us to pinpoint hotspots and understand disease incidence. They also let us analyze outbreaks across both space and time, which is critical for understanding how transmission evolves and not just where it starts. By integrating epidemiologic and genomic data, transmission networks give us much more fine-grained view of who may be connected to whom beyond what either data source can show alone. All of this directly supports outbreak-specific intervention decisions like where to focus contact tracing or infection control efforts. And finally, these insights don't just help with the current outbreak, they also inform future prevention and preparedness strategies. So now let me show you how MicrobeTrace transmission networks come to be. The first image shows nodes of an outbreak, where circular nodes represent people and squares represent facilities or locations. The second image shows person-to-place links. I've done that by adding epidemiological data, such as an edge list, that we've gathered through investigation. So now we can see the links appear. In the last image, we see genetic links. By adding genomic data, we can start to visualize which groups may have similar genetic sequences. MicrobeTrace is customizable, allowing you to layer other information, such as sizing the nodes by how many links they have, or even coloring the nodes by outcomes. This gives you a better picture of how transmission patterns form and connect. So, what type of data is needed to create a MicrobeTrace transmission network? Well, first, you'll need a line list, where each row represents a case or a facility. The line list may also include relevant demographic or epidemiological data. Second, you'll need linking data, in this instance, an edge list. This list defines relationships between individuals, such as who visited which facility, who was exposed to who, or had contact with who. To take full advantage of MicrobeTrace, you'll also want to add some sequencing data, whether it be Newick files, raw sequencing, SNP matrices, etc. So now I'm going to show a demo. If you want to follow along, go to Google and type MicrobeTrace. In the second tab, go to the GitHub link and download the files from the repository above. Notice that when we start loading the files, we're not uploading the files onto MicrobeTrace. So with that being said, let's get started. So before we jump into MicrobeTrace, I do want to go over the investigation that we'll be looking into today. So here is the background. You work for a state health department that has identified several cases of carbapenem-resistant Acinetobacter baumannii. Based on your state's investigation guidelines, you'll need to investigate all the facilities that an identified case has visited in the last 90 days. Additionally, if there are known roommates at any location, they are also to be tested. If a roommate tests positive, then point prevalence screening is conducted at the location to identify the extent of the spread. Your assignment is to explore the Okay. Anyway, so from using that, I'll give you a quick overview of the investigation. Now, this is a lot to read over, so I'll summarize it. As you can see, this investigation involves multiple facilities, along with multiple cases and multiple antimicrobial genes. And I'll explain the node list now. So if you remember from a node list, the definition of a node list, each row represents a case or a facility involved in the investigation. So as you can see, each case here and each node here, or each facility here, shows up in the investigation. So right away, from this node list, we can identify whether it's a case or a facility, but MicrobeTrace can't do that. So in a separate column, I categorize each row, whether it's a case or a facility. Then I further specify what AMR gene that case may have and what type of facility it may be, such as a long-term care facility, a hospital, or a clinic. I also add other relevant data, such as data diagnosis or coordinates of that case. It's important to note that you can add other relevant data than what you see here. Also, it's important to notice that these nodes are unique in name. You can't have a node with the same name appear twice in the node list, otherwise MicrobeTrace will throw an error. Now, we'll talk about the link list, or the edge list. So as a reminder, an edge list is the linking data. In this instance, it defines the relationship between person to place link. So here we see case 1, or person 1 visited facility D, and I further categorize them if they had visited this facility before, after, or during their date of diagnosis. I also add their start and end dates of visiting this facility. Now, with all that data, we can start into MicrobeTrace. So this is the page that you'll see right away when you get to MicrobeTrace. And to drop it and to add the files, you literally just drop them in. And I'll start out slow by showing you the node list first and how we customize that. So here we see that right away, MicrobeTrace has identified this Excel spreadsheet as the node list, as you can see here. If this had been incorrect, we would have just chosen the correct file type by clicking on the tab here. It also identifies what column you've chosen as your ID. It does a pretty good job of this, but if it was incorrect, you would just click on the drop-down menu and click on the correct column name from your spreadsheet. In some instances, others may add the sequence on the node list itself. I haven't done that in this instance. And then to launch this, we'll click the launch button. So right away, we'll see that here are our nodes, and we'll see down here the number of nodes. As you can see, we have 22 nodes, but these are pretty hard to distinguish. We can't tell what is a case and what's a facility. But if you hover over them, you can see the ID name. So to customize this, we'll click this cog button here and go to Nodes, and under Labels, we'll click ID. Notice as you scroll down the drop-down menu, your column names start to appear. So if I click Node, it would also be the same ID. Now you can also move the IDs, the orientation of the IDs, left, right, top, bottom, or even in the middle. For right now, I'll stick to the top. Now, to further distinguish them, we want to change the shape of them based on their node type, whether it's a case or a facility. So if you remember from the spreadsheet, we chose that name of that column was called node type. So right away, we can see MicrobeTrace has changed the shape of the nodes and we also see the number of each node here. So we can see there are 16 cases and 6 facilities, and the cases are represented in circles while the facilities are represented in squares. But this is still pretty hard to distinguish since everything is blue. So then we want to distinguish them further by using colors. And if you remember, in the column name that I used for that specificity was specificity. And right away, you can see that the nodes change colors based on the node type. So, and it also gives you a frequency. So there are 5 cases with OXID23 genes, and there's only 1 hospital and 5 long-term care facilities. So that is one way to customize the nodes. But now we'll want to look at some linking data. So we'll want to add that information by clicking Files, clicking Add Files, and clicking Link File In. And right away, you can see that it identifies it as a linked list. And it already has identified the columns used for the person place links. And we'll click Update. So now you see that these nodes are linked, but we've also lost the customization that we've done earlier. But that's not a big deal. We can just click through this again. So it's pretty messy, so you can always move these around. I like to start out by moving these nodes out first. And then I like to take the nodes, and if they only have one link, I like to place them out on the outside. If there are nodes with 2 links, I like to place them in the middle of the 2 facilities that they have visited. You can also choose multiple links by clicking on one and clicking the Control key and clicking the rest. So now this gives you a better idea of how transmission is occurring. And you can also see that these facilities are mostly contain patients with OXID24 and while these facilities have patients with OXA23. And you can also see that this facility also has patients with OXA24 and 23 genes. But the links still don't give us a lot of data other than just showing us what facilities the cases may have visited. So to add some more data to these links, we can color them. We can also color code them. So if you remember from the link list, I defined whether they visited the facility before, after, or during their date of diagnosis. So we'll color the links by going to this cog, clicking on links, going to show colors, and then clicking color links by, and that column name was called exposure. And you can also change the color of the nodes or the links by clicking on the color itself. And if you want to change a different color completely, you can just slide it down here and then choose a different hue on the color shade. And right away, you can start to see where the cases were before or after or during their date of diagnosis. But this still doesn't give us a lot of information other than when and where they visited. Now we'd like to add some genetic data to see how closely related these cases are. So we'll add another file. And we'll add our SNP matrix. Now notice that when I added the SNP matrix, it didn't select the correct tab right away. And that's not a big deal. All we have to do is click the tab here and change the file type. And then we click update. But again, we see that all our customization disappeared, but we get to see the different links that exist between these nodes, the genetic links and the demographic links. Now to add our style back, I can definitely do what I did previously and go through the customization, or I can add a style that already has the different settings. And to add that, we'll go to styling, and then we'll choose a style by clicking browse. Now this is a style that I have already created, and it'll seem familiar to you. Now, in order to create a style file, so you can just drop it again without having to redo the customization, all you have to do is go to File, you'll click Save, and this time you'll click Session. You'll click Style, and then you'll name it and click Save. So next time when you want to use the same style, meaning that you want to use circles as the cases, facilities as the squares, and use the same colors for your links and everything else, all you have to do is click Settings, go to Style, and then click Browse, and then choose your style file. So now you can see that it is a bit messy with the links. Now, right away, we can see that, as we had suspected before, that facility B, C, and A, they had some of the same patients, and you can see that there's a lot of relatedness among these nodes. And also, you can change the SNP threshold to show less or more links based on the SNP threshold that you type in. So in order to change the SNP threshold, you'll go to Settings, You go to filtering, and right now the default setting is for 16 SNPs. But in a paper that I've read, we've discovered that sometimes the 2 SNP threshold does a better job of distinguishing the same source outbreak, but that's not always set in stone. So right away, we can see that the number of links decreased just by observation here, and also with the number of links shown here. And still, our hypothesis seems to be correct that this data supports that there is definitely some propagated outbreaks occurring between facility B, C, and A, and the same thing for facility D, E, and F. Now, like I mentioned before, MicrobeTrace can also show other things other than transmission networks. It also shows phylogenetic trees. So if you click on the phylogenetic tree, you can see the phylogenetic tree created by the SNP matrix. Now, this is not always correct. Sometimes you do need to upload the phylogenetic tree itself to get a better view of the correct phylogenetic tree. Other views that we have include table information of your cases. So you can see the case name and the clusters in degrees, and I think there's other information as well. And you can select the columns that you want to show from your data that you've uploaded. Other views that you can look at are the SNP heat matrix seen here. Now this is pretty hard to read, but I think there is a way to show a better job of the phylogen of the SNP matrix by showing the names. So you go to SNP, you'll go again to the cog, and then click show labels down here. And there is a current bug with MicrobeTrace, and sometimes the facilities show. But I've already reported that, and they'll be fixing it soon. Other things that you can do is that you can take the views and align them together. So if you want to show the phylogenetic tree and the SNP matrix, you can just drag it into work. You can show them side by side. For example, if you want to show the table with information and the SNP heat matrix. And for the view, and for the map, which is probably the most interesting, we can go to the map, we'll go to the cog itself, we'll choose the longitude and latitude from the spreadsheets that we uploaded. So for these outbreak investigations, especially for carbapenem-resistant organisms, especially when the cases usually take place at long-term care facilities, hospitals, everything is kind of one in location. So it doesn't give a lot of information, but let's say that you were investigating like COVID outbreaks or measles outbreaks, this would probably serve better in those outbreaks and rather than these carbapenem-resistant organisms outbreaks. All right, well, that concludes my presentation over MicrobeTrace. If you guys have any questions, I'll be happy to answer them.